introduction. First of all, I would thank to organizers of this training, especially Professor Tanveer Saab, for inviting me to deliver a lecture in this training program. So before I come to this, my presentation that is on techniques in mushroom culture preparation and their preservation, I would first like to tell you about the culture, what is basically the culture of mushroom. So those who are aware about the mushroom cultivation, they may be knowing that they know spawn and they know compost. So spawn, what do you know about the spawn? What is basically the spawn? You see it in the form of seeds, and you see it like weed grain. Basically, it is not a weed grain. It is basically the culture of mushroom that is grown on that weed seed. So because the culture of mushroom is very small, we cannot carry it. So weed grain act as a carrier for this mushroom culture, and this act as a medium for the growth of that culture. So that means before spawn, we need some culture in order to prepare that spawn. Okay? So that is my presentation, how we make that culture, what are different techniques that we use for the preparation of that culture, and then we use that culture for the preparation of spawn. So this is basically my lecture. So coming to my lecture, as Dr. Tanveer Saab has already discussed about the mushroom, that he told you that it's basically a fungus. Now you know it's a fungus. So in case of fungus, we have a basically, as again Tanveer Saab has told you, that it is like the spore of a fungus is like a seed of a plant. So basically the fungus growth starts from the spore. Okay? So once the spore germinates, then it forms a mycelium, which is a vegetative part of the fungus. It is again like a plant has a vegetative part, and then it has a reproductive part also. So vegetative part of the fungus is mycelium that you see in the slide. I will show you. And basically this mycelium is generated from the spore. When the spore is germinated, then this mycelium is formed. So why I am showing this slide, here you will come to know which part of the fungus, which part of the mushroom we can use for the preparation of culture. So here you see, these are the spores of the fungus, and we, in case of mushrooms, most of the mushrooms, we call them bacillospores. As Tanveer Saab told you that we have another mushroom that is mortilla. That forms an ascospore, but most of the other mushrooms, they form bacillospore. So it starts from bacillospore. Once these bacillospores germinate, they will form mycelium. Okay? They will form mycelium, and this immediately the mycelium that is formed after the germination of spore, we call it primary mycelium. Okay? You remember this thing? We call it primary mycelium. So this primary mycelium is for a short period of time. So when this primary mycelium will find another primary mycelium which is compatible with this primary mycelium, they fuse and then form a second type of mycelium that is called as dikaryotic mycelium or secondary mycelium. So why I am telling this? Most of you may be having an apple orchards in your orchards, at your place. So in case of red delicious variety, we need, for pollination purpose, we need another variety. That thing you know. So similarly, in case of this mushroom, once the primary mycelium is formed, it will not form a secondary mycelium, it will not form a fruiting body. It is only the secondary mycelium that forms the fruiting body. So how this secondary mycelium is formed? It is formed once these two types of primary mycelium will fuse, which either one will donate its nuclei to another mycelium, then the rest of the mycelium that will form, which is the secondary, which we call as secondary mycelium or dikaryotic mycelium, it will be having a two nuclei. So this mycelium only, which we call dikaryotic mycelium or secondary mycelium, it is only this mycelium that will form this type of fruiting body that we call as mushroom. So otherwise, if this mycelium is only there, this primary type of mycelium, it will not form a fruiting body. It will form a mycelium, but it will not form this fruiting body. Because your mushroom is composed of secondary mycelium. It is only formed by the secondary mycelium. So this thing you know. Now let us see how we can use this mycelium or spores for the preparation of mushroom culture. In general, because we have so many fungi. We have some beneficial fungi, we have some harmful fungi also. So, do you know any fungi which is harmful to us, which is harmful to humans? Because fungi there are so many fungi. Some are beneficial, like mushrooms, they are beneficial ones. And we have harmful fungi also. Like fungi which cause disease in plants. You know ना, आपके पास तो भाग है ना घर में, तो आप जानते हैं कि वहाँ पे कुछ ऐसे fungi भी हैं जो plant में disease भी करते हैं ना. They are harmful ones. And this type of mushroom is beneficial one. And we have fungi which need living host for their survival. They cannot take nutrition from dead material. They always need living host for their nutrition. So that type of fungi, they are harmful because they need their nutrition from the living ones, so they have to parasitize the living host in order to take the nutrition. But these type of fungi, like mushrooms, they can take their nutrition from dead material. Okay? So they can use this dead material and then they absorb these nutrients. Because you know fungi, they don't have chlorophyll. You know? Like plants, they don't have chlorophyll. They cannot make their own food. So they have to depend on either on the living hosts or dead material to take their nutrition. So they take their nutrition from the dead material. So in general, fungi, when we use a fungi for their culture preparation, we can use their mycelium that is formed after the germination of spores. Either we can use mycelium or we can use spores. And in case of fungi, we have two types of spores because most of you are well qualified and most of you have a science background. You may be knowing that fungi is having a asexual spores as well as sexual spores. So that means we can use mycelium for the culture preparation, we can use asexual spores and we can use sexual spores. But in most of the mushroom fungi, we mainly have a mycelium, then we have a sexual spores. Like you know white button mushroom, in case of white button mushroom there is no asexual spore, there is only sexual spore, and that sexual spore is basidial spores, you know? That we call as basidial spore. It's basically sexual spore. I will not go into that detail. What is asexual spore? What is sexual spore? So that means, in case of mushroom fungi, we can use mycelium for culture preparation. We can use sexual spore for culture preparation. Now, what are different methods, different techniques by which we can prepare a culture? So when we use tissue or mycelium of a mushroom or a fungus to prepare a culture, that is called as tissue bed method. So when we use mycelium of a mushroom to form a culture, that is called as live tissue bed method. And when we use spore of that mushroom to form a culture, that is called as spore print method. And in case of spore print method, again we have two types, that is mixed spore method and single spore method. So before preparation of any culture, now you know we can use spore, we can use mycelium. But this fungus needs some substrate, some medium in order to grow. Now it cannot grow as such. It needs some substrate, some medium, so that it can grow. And what is that medium for when we prepare a culture? We call it potato dextrose agar. There are other mediums also, but mainly we use potato dextrose agar. And what is this potato dextrose agar? As the name suggests, it consists of potato, then dextrose is one chemical for nutrition, and then we have agar. Agar is some material which solidifies the material. If we don't use agar in that material, it will be in a liquid form. But we want in a solid form this medium, so we add agar that solidifies it. How it solidifies? Because at low temperature, below 45 degrees Celsius, this remains in a solid media, and if we give it 45 degree above temperature, it will melt and it will be in a liquid form. So agar we are using just for solidification. Now we have other medium also, but mainly we use potato dextrose agar, so you can use potato dextrose. It is a simple medium. You can simply bring potato from the market, and you can bring dextrose. It's not so much costly, and you can use agar in order to make this medium. Then this is first method, live tissue bed method. If you are using any tissue of the mushroom, that means that tissue will be consisting of what? It will be consisting of mycelium. है ना? मैंने आपको पहले ही बताया था कि mushroom is made up of dikaryotic mycelium. जो मैंने पहले आपको कहा था, there is primary mycelium, two primary mycelium fuse, they form dikaryotic mycelium, and from that dikaryotic mycelium, tissue is formed. But inside this tissue, inside this mushroom, there are spores also. If you have seen anybody has seen this this button mushroom, आपने देखा किसी ने button mushroom? So initially it is in a button form, है ना? Close होता है, button form में होता है. But later on it gets exposed. Umbrella खुल जाती है ना? तो उसके नीचे अगर हमने देखो ना अम्ब्रेला के नीचे देयर आर गिल्स ऐसे बने होते हैं ना गिल्स, तो उन्हीं गिल्स में इसके स्पोर्स होते हैं. But here we are not using these spores, we are just using the tissue of that mushroom in order to make the culture. So what is the technique of doing? इसके बाद शायद आपका practical भी है तो उसमें मैं दिखाऊंगा. They may be having some mushrooms. I will show you how to take the tissue from this mushroom. Basically what we do, we cut the, because you know mushroom is having the two parts. एक जो stem होती है ना इसकी base की, और दूसरा होता है umbrella shape का, है ना? तो जो stem है इसकी वो हमें काटनी इस तरह से. Vertically we have to cut the stem. Then we take the tissue where the stem and the cap join. ठीक है, जहाँ पे उनका joint होता है ना stem और cap का, वहाँ से we take tissue from that part and then transfer that tissue on medium which we have already prepared. जो मैंने आपको कहा in this case it is potato dextrose agar. और उस potato dextrose agar को हम कहाँ पे transfer करते हैं फिर media बना के? Basically, when we prepare media, we then sterilize it. It should be all sterilized, है ना? And that media we sterilize in autoclave. That will be a practical part of it. वो आपको सब दिखाएंगे. तो we need autoclave to sterilize that medium, and then after sterilization of that medium, we pour that medium into एक गिलास में होता है उसको हम पैट्री पैलेट बोलते हैं, गोल होते हैं. उसमें उसको pour करते हैं. Then we take this tissue from this mushroom and then we transfer that tissue on the center of the पैट्री पैलेट, उस मीडियम के सेंटर पे रखते हैं. Then after that we incubate that पैट्री पैलेट. Why incubation? Why do we incubate this pellet? After, in order to provide an optimum temperature to the mushroom, है ना? अभी आपको तनवीर साहब ने कहा था mushrooms need, most of the mushrooms need 25 degree Celsius temperature for their vegetative growth. तो वो इसको optimum temperature मिल जाए. In order to that we keep it in a BOD. BOD एक कैबिन होता है जिसमें आप you can give a optimum temperature for the growth of this fungus. Then उसके बाद आपने इसको BOD में रखा तो then you will see this type of growth formation in the pellet. This type of whitish growth. And this we call as, this type of growth we call as culture of the fungus. है ना? Basically this lecture is about how to prepare a culture. So this is how we prepare a culture of mushroom. And this is a method from tissue, which we call as tissue method. Then there are advantages of this method also, and there are disadvantages of this method also. So what are the advantages? It is easy. We can make a culture from any part of the mushroom, but preferably it should be from joint of this stem and cap. Then cultures can produce fruiting body. Now I will ask you question. जिसने ये मेरा lecture अच्छी तरह से सुना होगा ना? Why I say कि this will give a fruiting body, है ना? There are some other methods which will give a culture, but they will not give a fruiting body. ठीक है? ये आपको fruiting body क्यों नहीं देगा? ये दे सकता है. ये वाला जो culture है ना, इससे जो आप mycelium बनाएंगे, उस culture से आप फिर सुपान बनाएंगे. That सुपान will give you fruiting body. But there are other methods like single spore method. They will not give you a fruiting body. क्यों नहीं दे सकते हैं? ये क्यों दे सकता है? Exactly. So this culture we have prepared from tissue, which is consist of a dikaryotic mycelium. And in the beginning of my lecture, I said it is only the dikaryotic mycelium that can form fruiting body. And this is true for some fungi, which we call as heterothallic fungi. Heterothallic fungi means they need a compatible strain in order to make a dikaryotic mycelium. ठीक है? But there are some fungi which are homothallic. They don't need this compatible strain. ठीक है? So this is their advantage. We can get directly fruiting body from this culture, and this is easy to prepare this culture. But there are disadvantages also. What are these disadvantages? Once we take a mushroom for culture preparation, we have to do it immediately, you know, within 24 to 28 hours. If we keep it like as such, and we after 24 hours or 28 hours we are doing this culture preparation, this mushroom may decay, and there can be so many saprophytic growth on this mushroom, and we will not get a pure culture. We may get some contamination. So this is disadvantage of this culture. Then second method. This was tissue bit method, where we get the culture from tissue of the mushroom, and then we have spore print method, where we get culture from the spores. You know, we have to harvest the spores from the mushroom. Then we can use these spores for the culture preparation. So spore print method is, I said कि we have mushroom, है ना, और उस mushroom के अंदर फिर spores होते हैं. We have to first harvest these spores from the mushroom. Then these spores can be used for the culture preparation. So for the harvesting of spores, there is one technique that is there. First you have to cut the stem part of the mushroom. Then you keep this cap, this on the clean paper. Then cover this paper and this cap with glass jar. Okay? So that it is not disturbed. So after 24 hours, when you will come, you will see all spores are harvested. They are on the paper. Okay? Now it is only the spore mass which is on the paper. Now this spore mass you can preserve for future use, or you can immediately use this for culture formation. So this is spore mass. You can preserve it, as I said. You can preserve it in a polythene bag for future use, or you can immediately use this for culture preparation. How we use this for culture preparation? There are different methods for culture preparation. Once you have this spore print, spore mass, then there are different techniques to use this spore mass for culture preparation. One is called as scalpel or streaking method. Here you just dip the needle in this spore mass, then streak the spore mass. जो मैंने आपको पहले दिखाया था पैटरी पैलेट पे मीडिया है. तो once you touch the needle with the spore mass, then you streak that spore mass on the surface of the medium. So after incubation of that pellet, you will get a culture of that fungus. Then second is paper rubbing. You have a spore print, spore mass on the paper. You can just rub this paper. Spores will fall on the media, and that will produce the culture. Then you have spore syringe method. In this method, what we do, we use this spore mass and we make a spore suspension in water. We drop them in water, then we make a spore suspension. Then with the help of syringe, sterilizer syringe, we pick some spore, this volume of this spore suspension, and inject that spore mass on the surface of media. Again, that you have to then incubate, and that will give you a culture. Then we have single spore method also. Single spore method means we are using a single spore to generate a culture. So how we will do it? We will again make a spore suspension in water, sterilized water. Then we will dilute it and spread this some part of this solution on the media. Then individual colonies that are growing there, they will be from a single spore, because we have already diluted the spore suspension. When we spread that spore suspension on the surface of the media, there will be colony growing, and these colonies will be from a single spore. You pick that single spore colony, and that will be a culture from a single spore, and that we call as single spore method. So again, this spore method has its advantages and it has also its disadvantages. So one advantage is viable cultural potential fruiting bodies can be produced. So if we are using mixed spore mass, then there are chances that we will get a fruiting body, because in case of mixture of spores, we will be having chances are that we will be having two compatible strains there, because it's a mass of spores. We will be having both compatible strains there. They will form a mycelium, first a primary mycelium, then they will form a secondary mycelium, and from that type of culture we will get a fruiting body also. But whereas it is a single spore method, if it is a single spore method, from single spore method we will get a culture, we will get a mycelium, but if we develop a spawn from that culture, we will not get a fruiting body. Again, reason is same, because once it is only a single spore, it will form a primary mycelium, but it will not find compatible strain, because it is only single spore. There will be no compatible strain, it will not form a dichromatic mycelium, so there will be no fruiting body from that type of spawn. So that is its disadvantage. But why then we use this single spore method to generate a culture? No. So why we are using this single spore method? If it is not going to give a fruiting body, then why we are using this single spore method? Why? Because it is for an R&D purpose. Tomorrow if you are going to start this venture as a big venture, then maybe you are doing an R&D part also, research and development part also. So for that person, sometimes you need a pure strain. So you can collect different strains and then cross these strains so that you can see which compatible strains, two strains can give you a strain which is giving a good yield or which is having some good characteristics. So it is for that purpose, it is for research purpose. Then this is culture, how we develop a culture by any technique, whether it is a tissue technique, whether it is a support technique. Then finally we use this culture that we have developed, this culture we use for spawn production. And for spawn production you can use the spawn that you might have seen, it consists of a wheat grains. So it is not only wheat grain that we can use. Different mushrooms we have different grains that we can use. We can use maize grain, we can use wheat grain. So commonly we use wheat grain. So then we inoculate this culture that we have developed. We inoculate it on the wheat grains. Then finally we have a supan. ठीक है? तो इसके लिए फिर क्या करते हैं हम? What is supan? Supan is, then we say it is a seed for mushrooms. Basically, it is a mushroom. It is a culture which is seed for the mushroom. But this culture needs a substrate. This needs a carrier. Only that we inoculate this culture on the grains, so that we have a culture, we have a supan, so that we can carry it and we can use it for mushroom production. So that is basically supan. So for again, because we are using wheat grains, we can not use wheat grains as such. We cannot use them for a substrate for culture. Maybe they may be already contaminated. If we directly inoculate this culture on the seeds, there can be have different types of organisms on seeds, and they will contaminate our culture. They will not give a pure culture. So for that, so this is the ingredients that we use for the culture, this preparation of supan. We use whole grains, that can be wheat grain, that can be maize grain, 10 kg. Then we use gypsum and we use chalk powder. Why we use gypsum and chalk powder? In order to maintain the pH and in order to avoid the clod formation. Okay, this is for that purpose. Then how we sterilize that material? Because we have to, as I said, it is already contaminated by so many organisms. So in order to avoid that contamination, we first prepare these wheat grains for inoculation. So how we prepare it? We first boil them, we make them soft. Then we mix all this gypsum and other things at the rate of 2% gypsum and at the rate of chalk powder at the rate of 0.5%. Then we fill these material in the bottles, which is the glucose bottle आपने देखी होगी ना, उनमें fill करते हैं. Then we autoclave that grain. क्योंकि उसको autoclave करने से क्या होता है? Autoclave क्यों करते हैं? Autoclave is for sterilization purpose. Autoclave is basically, it is like a pressure cooker that we use at home. So it increases the temperature, it increases the pressure. By that temperature and pressure, it gets sterilized. Whatever material we put in the autoclave, that gets sterilized. So after sterilization, we cool these grains. How we put them in this autoclave? We put these bottles in the autoclave, then we sterilize them at a specific temperature and pressure. Then we inoculate this bottle. Okay? This is how we inoculate. जो हमने culture बनाया था, it can be in the pantry pellets, it can be in this is called as test tube. It can be in test tubes, it can be in pantry pellets. From that, you take some bit of mycelium and then inoculate it on wheat grains. जो हमने अभी prepare किया ना wheat grains के लिए, तो उसमें आप इसको inoculate करते हैं. Then finally you incubate that bottle in a BOD. This is BOD. इसमें incubate करते हैं in order to provide optimum temperature. Then finally you get a supan. So now it is ready for inoculation in the compost, so that you will get mushroom. So this is story about how we make a culture. Then from that culture, how we inoculate that culture on any type of substrate. It can be wheat grain, it can be any grain, so that we get finally this type of bottle, which is we call as supan. Now second part of my presentation was, now we have prepared the culture. Okay? Now maybe immediately we don't need this culture in order to make supan. So for a period till we use this material, we need to preserve that culture, because it is a living thing. It is a living thing. It is growing on a media. So media finally it will exhaust. Once the fungus takes nutrients from that media, finally it will exhaust. And second thing is, there will be accumulation of toxic substances in the culture, and this will cause the death of the culture. So in order to avoid that thing, we need to preserve our cultures. And there are different methods of preservation of cultures. So there are two types of methods of preservation of any culture. It can be your mushroom culture, it can be any culture, bacterial culture, whatever you are interested in. So there are two methods basically. One is short-term method, where we can preserve our cultures for 2 months, 3 months, mostly 3 months, up to 3 months, depending upon the culture. Then we have some long-term methods that are for to preserve our cultures for up to 10 years also. So among the short-term methods, we have subculturing method, we have refrigeration method, then we have mineral oil method, then we have paper disc method also. And in case of long-term methods, we have cryophilization and lyophilization. So in case of short-term methods, first method that I said, it is subculturing method. What is subculturing basically? It is, you have a culture, you want to preserve it for 1 month, 2 months. Once the medium is exhausted, you immediately transfer that culture into new medium. Okay? So before the medium is exhausted and there is accumulation of some toxic substances, you immediately transfer that culture, bit of that culture on another slant, so that it will have a fresh medium to grow. So that will keep it live for 2 to 3 months. So once that new material is again extracted, before that you will again transfer it to new culture. By this way you can preserve your cultures for 1 to 2 months. Then there is refrigeration. What is refrigeration? You keep your cultures in a refrigerator, and in case of refrigerator, you know we have 4 degrees Celsius temperature. So lowering the temperature, it will slow down the growth of the fungus. So once the growth of the fungus is slowed down, so there will be, it will be preserved for a longer period of time. It will exhaust the nutrients less, and there will be accumulation of these toxic substances less. So it will prolong its shelf life, but it also needs then subculturing. It will not keep it for a more period of time. You need to subculture it again. Once it is refrigerated, maybe in case of simple subculturing method, you can keep it for 2 months, but by refrigeration, you can increase it for 1 or 2 months also. So again, it will need subculture. So there are advantages and disadvantages of this method also. It's a simple method, easy to recover the culture. Okay? No special technology is needed in, just you have to subculture it on the media. Then disadvantage is that while subculturing the culture, it may get contaminated. Okay? So that is disadvantage of this culture, this method. Then you have mineral oil method, one short-term period of preservation. So what we do in case of mineral method, so the culture that we have in a petri pellet or in a slant, you put mineral oil on the top of that culture so that there is no oxygen. Okay? You provide an anaerobic condition because your culture needs oxygen. Once you put mineral oil on the top of the culture, there is no oxygen supply to the culture, and the growth of the fungus will be stopped, and this culture will be preserved for more period of time. Then long-term method, as I said, one method is cryopreservation, and in case of cryopreservation, what we do, in case of refrigeration, it was 4 degrees Celsius, but as I said, at 4 degrees Celsius, we can have some growth, but here we store it at minus 190 degrees Celsius, and that is only possible in case of liquid nitrogen. We put them in a liquid nitrogen so that there is very much low temperature. At this point of temperature, there is no growth of the fungus, and this remains viable for a longer period of time. Then another method is, last method is lyophilization. In case of lyophilization, what we do is also called as freeze drying method. First we freeze the culture at minus temperature, very low temperature. Then we dry it. We dry it means we take all water, it's through vacuum, there is some special equipment for that. We remove the water from the culture, we dry it, then we can preserve it for a longer period of time. So these are some, but there are advantages of these long time methods and there are disadvantages. Advantages, we can preserve our cultures for 15 to 20 years, but the disadvantage is it is time consuming and it's costly method. Everyone cannot afford this type of equipment for preservation of culture. So this was all about my lecture. So if you have any query, you can ask, please.
सर मैं एक चीज़ समझना चाह रहा हूँ इसमें सर, जो भी सर इसमें स्पान प्रोडक्शन आपने कहा कि हम अगर कोई फार्मर इसको बनाना चाहेगा, तो मेरे ख्याल से मुझे लगा जो आप प्रेजेंटेशन से मुझे लगा वो एक फार्मर के लिए खुद से प्रोड्यूस करना थोड़ा मुश्किल है.
हाँ, तो इवन पहले आपको कहा ना, at a small scale you take this span from some source, है ना, किसी भी इंस्टीट्यूट से आप इसको span को ले सकते हैं. You don't need to form this culture, है ना. It is only in case of when you start that big venture, है ना, वहाँ पे you are interested in, जैसे मैंने आपको कहा कि आप खुद अपनी स्ट्रेंथ कोई डेवलप करना चाहते हो, है ना, तो आप कहीं से भी कोई मशरूम लाया आपने जो आपको लगा कि it is a good mushroom, it is इसका जो कोई भी क्वालिटी पैरामीटर अच्छा है, तो you want to make a culture from that. जैसे आप देखना कि जैसे आपके पास आर्चर्ड है, आप कहीं पे जाते हो तो आप देखते हो कोई अच्छी सी वैरायटी किसी प्लांट की ना, तो you bring that variety and propagate that variety. तो इसी तरह से अगर आप इस चीज़ में ज़्यादा डीप जाएंगे ना, So sometimes you will see some mushroom, कहीं से भी देखा आपने, तो you want to use that mushroom as a spawn, तो first we have to make a culture. तो उसके लिए आपको ये culture पहले बनाना है. Then you can make a spawn. तो small scale के लिए just you get a spawn from any institute, then you can use that spawn for mushroom production.